Yes, so I have a question about the microbiome. So you have hundreds or whatever, thousands of microbes living in you. How do you actually go and, other than separating out the 16S ribosomal, how do you then attribute all the other sequences that are there to a particular genus? Or do you?
There are reference databases, so when you do metagenomic sequencing, which is what I assume you're asking me about, you're looking for the other functional genes that are part of the genomes that are there. One of the weaknesses right now is that we don't have very many genomes that we've fully sequenced. So it's growing though, and there's been great progress made by the HMP to create those known genomic references. But that's why so many of the genes at the functional level are not identified at this point, is we don't have a great, the reference base is a little lacking still.
Well, I just wanted to make sure that I wasn't really missing something, because in terms of what we do in a practical manner, the amount that we actually can identify and associate is relatively small.
Do you mean with the human genome?
No, the human genome's fine.
Yeah.
What I mean, so what I mean is the microbiome that's associated with the human genome.
Yeah, so that's one of the differences, I guess, between human research and the microbiome research, is that with the human you have one scaffold, and with the microbiome in the gut anyway, you can have up to 1,000 different scaffolds. And then the synteny is different, so how the genes are actually arranged within the genome are not the same. And as you were showing earlier, there were differences in just 2 strains of organisms and the complements of genes that they may have. So it's complicated. So that's why we rely on bioinformatics so strongly.
Yeah, let me just expand the way you would do. So you can work it at various levels depending on your resources. 16S gives you the quickest and the least expensive. The other end of the scale, as Meredith indicated, is whole metagenome sequencing, where you sequence very deep to get very good coverage of most of the organisms that are there. If you care about only particular individual organisms, you can just do PCR assays for the individual organisms. And there's also sets of, sort of like the 16S, where you've got different levels of resolution for different core genes across the microbiome, and you can PCR those out and sequence those and get species level views of those organisms.
One thing to that as well. So the data that I showed that was illuminating all of the different butyrate producing pathways, that paper actually has created sequencing primers which are geared towards those specific pathways of interest. And that seems to be the way that things are going at the moment. So.
Thank you.
Dr. Newman, I want to ask a related question in this area. So I was fascinated by this observation that these people in the Netherlands have made, that you can take somebody with a healthy teenager or college-age student and transplant their bacterial microbiome into the unhealthy metabolic syndrome person and basically lower their insulin resistance and have them become a healthier person. And what they attribute to is the super donors bacteria. And of course, you can do the same thing in a mouse, in a notobiotic mouse. But what happens is everybody kind of slips back to normal. So you have even the notobiotic mice, you don't see them carried out longer than a couple of weeks. And generally speaking, they're probably coming back to where they started, just like these people who receive the bacteria eventually need another transplant. So to me, it argues for a couple of things. One is that we have to know what the host genome is. And the second thing I just wanted to ask you is, how far down do you think we're going to have to go to the subspecies to understand who to actually transplant? Is that actually possible, do you think?
So the work that was done actually at WashU when Sarah Highlander was there was really focusing on individual species for managing the C. difficile situation in antibiotic-treated humans, the result in the proliferation and the in many instances near fatal infections with Clostridium difficile. And it looks like with just from what she was presenting in a seminar on this topic that it was down to 3 individual or 4 individual strains of lactobacillus that brought about rapid remission of symptoms. So in the case of C. diff infections, it's totally a consequence of antibiotic interventions. So over time, and maybe Meredith can have more information on this, over time, after a severe episode of antibiotic treatments, the microbiome returns to something resembling its former state, but it takes a bit of time to achieve that. I've heard presentations where you do dramatic alterations in diet and you get within days dramatic modifications to your microbiome. So you can perturb the microbiome as long as everything stays the same. It'll come back to the way it was over time, and so the transient remission of whatever symptoms that you were alluding to, I think, is a consequence of a transient proliferation of what you put there, competing with everything else that's there. As you alluded to with the genome and the immune system status, which is not completely a consequence of the genome, it's the consequence of your whole experience in living on the planet and what you've been challenged with. All of these things are contributing to what's going on there.
Hello, I'm Dr. Alan Brown. I'm from Maine. I'm a pediatric bariatrician and a pediatric surgeon, and my question's for Dr. Convit. There, I noticed Dr. Wren's on your list of your colleagues at NYU. Have you had the opportunity to take any of the adolescents who've had a significant weight loss and redo some of your MRI, your retinal arteriolar measurements, and/or your sleep studies to see if there's effect on those parameters?
No, that hasn't been done. So I'm collaborating with folks out of the adolescent labs, which is a longitudinal follow-up of kids, but that's only doing cognitive longitudinal evaluations, which is too bad because they didn't actually. I'm in the process of trying to get some funding, and it looks like it's going to be successful this time around, given the previous reviews, to actually track people longitudinally before and then after bariatric surgery, to then measure a whole bunch of stuff, not only in brain, but also inflammatory markers and doing detailed insulin sensitivity assessments and so forth, to really try and develop more causal-like models that one can do now with fancy statistics, even without randomizing people into surgery or no surgery. You can use models that get you fairly close to causal. So I'm really looking forward to doing that, but there is very little information. There is information that cognition improves with bariatric surgery and massive weight loss, but what the mechanisms for those are is unknown.
Hi, Rob Waterland. I have a question for Dr. Convit also. So your data suggests that overweight and poor fitness lead to impaired cognitive development in children. And so I'm wondering, with the increase, the secular increase in adiposity in the population in the last few decades, including among children, is there data on, is that affecting academic performance of kids?
Well, we know that certainly if you look at the big numbers, like that study that they did in New York City, it does seem to be affecting academic performance.
But are there any longitudinal data at the nationwide level?
Not that I'm aware of. So some of the NHANES data has some educational information, and they have been tracking kids, but I'm not aware of what that data shows. Now, you have to bear in mind that the differences in cognition that we're measuring in the lab, although there may be statistically significant, it's only about a 10% difference. So it's not like the kids who have dysregulation, metabolic dysregulation, are performing in the abnormal range. They're just performing less than their potential is the issue. So it's really an important issue, particularly when I explain it to parents, that helping their kids get fitter by incentivizing them, by bribing them, paying them, doesn't matter what. You can pay kids to play sports. So, and they will eventually like it. And that's going to cost you a lot less than dealing with all the complications of not playing the sports. So.
Well, please join me in thanking our Just a couple of concluding remarks. Well, I guess we now have clinical application. We'll pay all of our children to be physically fit, and everything will be well and reversed, and we'll make sure that our hippocampus has appropriate volume. I want to first thank you for coming today and for your attention. It's certainly been a stimulating day. And T.S. Eliot said that in our beginning is our end. And so in my concluding remark, I'd like to take us from this point of the end of day one and just offer just a couple of remarks. As we started, we know that DNA is a code where context matters. And what you've heard throughout the day is that context could look like maternal diet, paternal diet, prior generations and their experiences, the changing microbiome. These can create marks measured in various ways through various research designs, exploring causation, that these marks may vary by tissue, age, and gender. And tomorrow, we will take the complexity of that and talk about how we can still move forward. What are the next steps, and are there opportunities for clinical application? So thank you again, and we look forward to seeing you tomorrow morning at 8:50.